Hello everyone. First, I would like to thank the organizers for offering me the opportunity to share our experience in Luxembourg on the genomic mapping of SARS-CoV-2 using deep sequencing. I'm presenting this work on behalf of the Microbiology Department in the National Health Laboratories. Since the emergence of SARS-CoV-2 in December 2019, the coronaviruses, which are enveloped positive sense single-stranded RNA viruses, took a center stage. We knew of 6 coronaviruses causing human infection, but the question raised was, which coronavirus are we facing? Is it one of the so-called seasonal coronaviruses causing up to 15% of common cold cases worldwide? Or is it going to be one of those coronaviruses causing severe diseases similar to the SARS and the MERS? So a lot of efforts had been put in place to understand more about the nature of this virus as much of the nature of the disease that it's causing. The new coronavirus was successfully sequenced in January 2020 in China. The virus appears to have close to 80% genomic similarities with the 2003 SARS-CoV. This was a breakthrough for the molecular diagnostic applications because we were able to identify the conserved regions on the genome that could be a potential target for molecular diagnostic assays. And indeed, there were 4 main targets that are currently used in all of the RT-PCRs for diagnosis of SARS-CoV-2 currently, namely the N gene, the E gene, the S gene, and the ORF1. Microbial genomics plays a major role in understanding of the epidemiology of emerging pathogens. Hence, there was a need for a protocol that allow us to obtain whole genome sequence data for SARS-CoV-2. The Arctic protocol came up with an amplicon sequencing approach that uses a pool of primers that target different regions of the SARS-CoV-2 genome, and when put together after sequencing allow us to have a full picture of the full genome. Locally in Luxembourg, we had adapted that protocol by decreasing the number of primers needed to up to 51 primer pairs and increasing the fragment that we are sequencing up to 900 base pairs with overlap of 200 base pairs in between fragments. This actually is a good tool for future studies on the diversity and also when it comes to haplotype reconstruction. With that kind of protocol, we had an optimal coverage of the SARS-CoV-2 genome, being able to nearly full genome sequencing. And what we have seen, like a lot of our colleagues in different labs, that the viral load has an implication on the quality of the sequencing that you can get. So with a CT value of around 29, we still can have a high quality full genome sequence. When the CT value go higher, we still can call the clade up to 32 CT value. However, higher CT values had been linked with failure to obtain the whole genome sequence, which is a known thing when it comes to sequencing applications. So once we retrieve the raw data from the sequencer, we proceed with the data analysis. For that purpose, we had developed an in-house bioinformatic pipeline that is based on a mix of open source software and locally developed scripts. The first step in this pipeline is the quality check, where we make sure that all of the sequencing outcome is valid to be processed in downstream analysis. Then we start the data cleanup, where we only retain the viral data and we try to exclude any human reads or primers that were introduced during the sequencing process. Once we obtain the viral data, we map that data against a reference sequence. In this case, we use the Wuhan sequence to obtain what's called a consensus sequence, representing the full genome sequence of the SARS-CoV-2 in question. Then we take that consensus sequence and we submit it to an online tool provided by the University of Glasgow. Its name is CoVGlu, where it offers you a list of all of the mutations and deletions and insertions that are within your full genome sequence when compared to the reference strain. So once we have this kind of data, we can then proceed in the data analysis, and we can also use the consensus sequence to plot a phylogenetic tree to see the relationship of this sequence to other circulating viruses in Luxembourg. We wanted to issue a kind of a medical report and give the whole genome sequencing data a clinical relevance over here, rather than its only application from the epidemiology point of view. So we had performed an extensive literature review to find the reported mutations of clinical significance. We identified 8 mutations that were reported recently that confer either higher infectivity, higher severity of the disease, or even improve the viral fitness. However, I would like to highlight that the data I'm presenting today wouldn't include the recently reported major deletion of the SARS-CoV-2 genome that has an effect on the severity of infection. A recent article reported in Lancet. This is one of our earliest phylogenetic trees that we produced to describe the circulating variants in Luxembourg, plotted here in brown circles. As you can see, we had plotted representative sequences from European countries, and as well as one reference sequence from China. We can see the clustering over here with Austria and Luxembourgish variants, and we can see as well another cluster that is shared between UK and Germany, Luxembourg and Switzerland. And we can say that there is epidemiological connection as well among these kind of samples. However, we are setting up a phyloepidemiology study with our colleagues in University of Luxembourg to understand better the transmission networks in Luxembourg. Back to the 8 mutations that we identified of clinical significance. When we studied the viral sequence data that we obtained in the period between March and May this year, we have identified 2 mutations that are circulating in the Luxembourgish variants. So the first one is D614G, which is well described in the literature to confer higher transmissibility and higher fatality rate, while the other mutation, which is B323L, is less described, but it is mentioned that it confers higher transmissibility. Maybe that's also related to the fact that it is located on the polymerase gene. We also describe the percentage of the different lineages circulating in Luxembourg, and you wouldn't be surprised that it's mainly the lineages that are present in the European region. So the dominant lineage is the B1 in Luxembourg, with only 2 cases mainly from lineage A2. One of them is a returning traveler from Spain. So we all agree on the importance of the microbial genomic in the SARS-CoV-2 era, either from public health point of view or from potential clinical application. And in the spirit of the COVID fight that we had been going through together, I would like to offer our expertise and knowledge to any interested party. And on that note, I would like to thank you all for listening.